All right, it looks like the trickle has stopped. And so I want to welcome you guys to our seminar titled A PhySSi Story. Introduce our speakers today. Blair Morrison is master's student in my lab. She's got her BS from the University of Alabama. And Alexandra Smith is a PhD student in my lab. She's got her BS from Vanderbilt University. And my name is Jeff. I got my PhD a while back. And that's all I have to say. This could be one of the most indulgent seminars you guys ever see at DISL. But the reason I'm excited to be a part of this and having Blair and Ali is because I haven't really given a seminar at DISL since my job talk in 2012. And we've changed what we do and the scope of what my lab does a lot since that first talk. And for students that are new to the program, we want to introduce you guys to the types of things we do in the lab and effectively who we are. So first, what's with PhySSi? Okay, we look kind of crazy. There's been some terms thrown around, cult-like. I'm not going to say who said that, but I've heard it. Well, we are the Phytoplankton, Sulfur, and Silicon Laboratory. And we use the Phi, the S, and the SI in our name. We were established in 2013 with our founding members, myself, Israel Marquez, and Eric Lackenmeyer, but we rebranded in 2019. We're a blended family lab resulting from the merger of the Krause and Keene labs. And we now represent over 20% of the periodic table elements that start with the letter S. It's pretty awesome. We're purveyors of fresh scientific ideas, scientific holiday cheer, which might be disturbing to some, and t-shirts, which are available for purchase if you guys like the swag that Ali and I are wearing. We have stickers, which, we got those too. But we want to tell you a little bit about the work we do, and we should basically start with who pays for it. Clearly, Dauphin Island Sea Lab pays for my salary, and that should be acknowledged, number one. A lot of the work that we do has been supported by the United States government, primarily the National Science Foundation, North Pacific Research Board, the United States Food and Drug Administration, but we have had projects in the past through kind of private research agencies like the Gulf of Mexico Research Initiative. And then we've also had a strong partnership with international collaborators, whether they're from Norway, the Research Council of Norway, or the German Ministry of Research, and even colleagues at King Abdullah University of Science and Technology in Saudi Arabia. The two projects to the bottom right of the slide I'm very excited about. The Mosaic project just finished. It was a project that basically put the Germans' crown jewel icebreaker in the Arctic pack ice for a year. And it just set sail and got back into Bremerhaven a couple days ago, and it had finished its year. And this was an exceptionally logistically complex enterprise to keep an icebreaker in a pack of ice for a year. And we're going to be getting samples from that eventually. In terms of where we do our work, we have past projects and current projects, mainly in the Northern Hemisphere, but thanks to the merger of the labs, we have representation in the Southern Hemisphere as well, including the Ross Sea and Antarctica. And these data from that study are still being written up by one of our lab members. You can see we do work locally, and we like to work in cold places or even in the ice. So I'll tell you why in a little bit. Who are we? Clearly the three of us, but we are larger. Our technician is Cassie Bates. We have a couple of PhD students. Allison Rellinger is our most senior, followed by Israel Marquez, and Randy and Ryan are our newest kind of members to the lab. MS students include Kyle Halstead and Blair Morrison, and then our recent alumni, Rebecca Pickering, who's now a postdoc in the University of Lund in Sweden, and Tara Williams, who's a PhD student in Pete Sedgwick's lab at Old Dominion. So this is who we are, and apparently they do a lot of shenanigans. As you can see, I'm not in most of these pictures, but it appears that the lab has a lot of fun. So let's get to some science. we're going to put the bio in biogeochemistry, and I want you to think about phytoplankton. For those of you that have had my bio-OSH course, this is what I like to call the big four: cyanobacteria, haptophytes, with the poster child being Aemiliana huxleyi, which is pictured in the skin and electron micrograph, dinoflagellates, and diatoms. And collectively, these are your main primary producers in the ocean. They're the main phytoplankton groups, and they dominate primary productivity in the ocean. The essential elements that they need for what they do include carbon, nitrogen, phosphorus, sulfur, and if you're a diatom, you need this extra element, silicon. Through the process of primary production, they're making organic matter, but again, for a diatom, they have to add that silica to make a shell of biogenic silica. Because these groups do so much primary production, we have this really strong coupling between the elements of sulfur and silicon with the biogeochemical cycles of carbon, nitrogen, and phosphorus. Now, we also do geochemical work. And so when you think about sulfur and silicon in particular, they're not just affecting things biologically, they're also affecting geochemical processes on long timescales. So for silicon, silicon weathering of rock is a really important process that's chemical, which is regulating Earth's climate. Why? Because it consumes CO2. And so this gives a cooling effect, that negative sign to global climate. If you want to go the other direction, we have reverse weathering, which is when silicates and reactive aluminosilicates are reformed to make new clay minerals. And the cool part about this is CO2 is a byproduct. And so because it's a byproduct, this process is called reverse weathering. It's not a strict reversal of the chemical weathering process above, but because it's adding CO2 to a marine system, which can eventually outgas to the atmosphere, this provides a positive kind of feedback to increase temperature in the ocean. And then with sulfur, sulfur processes, which Ali is going to talk about a little bit, they can release sulfur gases into the atmosphere, such as dimethyl sulfide, which can help stimulate the formation of clouds, which indirectly causes more albedo, shading of the atmosphere, which can cool. And Ali will discuss this a little bit more. So the format of this is we're going to bounce back a little bit. Blair's going to start, Ali's going to go next, and then I'm going to wrap it up. So right now, I'm going to relinquish to Blair. And Blair, you have to unmute yourself.
Yes, that is important. All right, let me share my screen. Okay, can y'all see it? Okay. Cool. All right. Well, thanks guys for coming to our seminar. We're going to talk first about our friends, the phytoplankton. And so as Jeff mentioned, our lab studies a lot of different processes on varying temporal scales. And so I'm going to talk first about kind of the smaller scale or like shorter time scale processes. And so our phytoplankton family here is Alex, he's on the top, myself, and Randy. So to start our phytoplankton narrative, I just want to kind of show you guys a generalized phytoplankton and planktonic food web. So on the left, we have our generalized phytoplankton. So as Jeff was saying, these include our diatoms, our dinoflagellates, cyanobacteria, and our coccolithophores. And then moving to the right, we have our planktonic consumers like our copepods, larval crabs, and larval shrimp, and then into our gelatinous zooplankton, and ultimately moving into our larger kind of fish. And so this is important because this, our phytoplankton effectively shuttle energy from single-celled organisms into more complex, multi-celled zooplankton, which ultimately go into higher trophic levels like our fish. And these kind of pathways are essentially the way that food webs are sustained, and ultimately our fisheries are sustained. And so this, though it is important to understand that this is an oversimplified kind of food web, this doesn't include the microbial loop, which is our like ciliates and other tiny, tiny organisms that actually consume a majority of the production that our phytoplankton produce. but for simplification, we're just going to talk about our larger, more charismatic plankton here. So zooming in on just one of the pathways that is in this, in our planktonic food web, we have our diatoms, which are one of our favorite phytoplankton here in PhySSi Lab, and our copepods. And so, as Jeff was saying, our diatoms, they essentially live inside of glass houses. So they uptake silicon from the water and they secrete biogenic silica. So they can form all kinds of really interesting patterns and spines and everything. And that's because our diatoms and our copepods exist in this evolutionary arms race because they are in a direct predator-prey relationship. It is kind of hypothesized that diatoms first started secreting shells to prevent predation from copepods, but then copepods were trying to get around that and they started developing jaw structures and teeth that could break the silicon shells. And so because this kind of evolutionary arms race happens, this structures a lot of the research that Alex and Randy do in our lab. And that process of the evolutionary arms race and those predator-prey interactions are really important ultimately in coastal oceans and in polar seas, as Jeff was also mentioning. So for Randy's work, she studies mostly the diatom physiological response. And so this idea is actually really interesting, the idea that you could scare them into growing thicker shells. And so diatom shell thickness can be affected by light, it can be affected by temperature, and the availability of silicon in the environment. But Randy is looking at if chemical cues can affect the thickness of diatom shells. And so she is currently working on some pilots of studying different chemical cue releases of cultured diatoms in our lab. For some of her cultures, she's going to expose them to chemical cues from just the presence of copepods, so the natural kind of chemical cues that they give off in the water. And then some other of her cultures, she's going to be exposing them to kind of this diatom death cue. So that would indicate that copepods are actively feeding in their environment. And ultimately, she's hoping to see an increase in silicification of the shells of the diatoms. And we study this by looking at the kind of concentration and change in biogenic silica, which you can see in these little bottles that are on the lower right. We developed them and it can tell us about the biogenic silica content. So by looking at this process, Randy ultimately hopes to figure out how these kind of chemical cues, how they're affecting the physiology of the diatoms, if there are any other cues that can affect their physiology, and how this interacts with physical constraints of the environment. So if diatoms were in a silicon-limited environment, would they still grow a thicker shell even in the presence of predators? And then this moves into some of Alex's work. So he studies more of the copepod feeding mechanics and dynamics of this side of the evolutionary arms race. And so just backing up a little bit, like we said, diatoms live inside of glass shells, and diatoms are really strong. Even though they're tiny single-celled organisms, if you scaled them up to the size of a coffee table, an elephant could stand on it and it wouldn't break, which is pretty crazy to think about. So even though, you know, they're that strong, how does a tiny little copepod crack open a shell and eat diatoms? So for copepods, they're incredibly choosy grazers. And some of the factors that they have to consider when they're figuring out what their prey items are going to be is they have to consider the amount of energy it takes to break open the shells themselves. And you can see some of the fracture patterns in these photographs. So these are stained diatom shells and confocal images of diatoms. So it takes a lot of energy to break through shells. They have to consider that. They also have to consider the nutritional value of whatever cell is within the glass shell. So, you know, we talk about a low energy kind of like a Snickers bar versus a hamburger. You kind of want to go for a hamburger, even if it takes a little bit more energy to crack the shell. And then the last thing that Alex has discovered is this idea of a food memory can be important in determining what they're grazing on. So if the food item is a novel food item, or if it's something that's constantly in their diet, that can affect the way that copepods graze. And so these are some of the primary characteristics that he studies, but he was also interested in seeing if secondary metabolites or toxins can affect the way that copepods graze. And that brings in our Pseudonychia diatoms. So Pseudonychia have thinner silicon shells, so they're theoretically easier to break open for copepods, but they produce domoic acid. And domoic acid is known for causing amnesic shellfish poisoning, and it's been linked to a lot of shellfish closures on the west coast of the United States. So this can be a kind of persistent toxin in the marine ecosystem. And so he wanted to know if that toxin would kind of affect the way that copepods are grazing on those cells, even if they're more common in the environment. And he actually found that copepods don't really seem to be affected by the domoic acid at all, which means that they can be a really toxic vector of moving domoic acid through the marine food web. But even stranger, he described this pathway in which copepods don't even need to directly eat the Pseudonychia to carry domoic acid into higher trophic levels. They can actually eat marine snow that aqueous forms of domoic acid have absorbed onto and then move it through the food web. And so this process, this paper recently came out, and this kind of sheds a new light on the way that marine toxins can be cycled and can retain inside of marine food webs. So again, we have our domoic acid that's being absorbed onto the surfaces of marine snow, and TEP, which is transparent exopolymers, essentially what cells kind of shed off just extra carbon and the sort. And domoic acid can move through the food web through direct grazing and also through these marine snow kind of pathways, which is really, really cool. And last but not least, I want to take us back to our larger food web that we started with. And so I said that the microbial loop has been excluded from this narrative so far, but it's important to note that microbes are still an integral part of this system. And so all of the kind of little bits and pieces and pathways in this food web can actually serve as vectors for moving microbes through a food web into higher trophic levels. And for my work, particularly the microbes that I study are Vibrio bacteria. And so Vibrio bacteria, a lot of us know of them through, sensationalized headlines of, like man's leg gets eaten off, by flesh-eating bacteria. But actually, those kinds of infections are very numerically rare, but they are important enough and severe enough to warrant a lot of research in the mitigation of risk of Vibrio. And so Vibrio, the two species that I study are Vibrio vulnificus, or VV, and Vibrio parahemolyticus, or VP. Vibrio vulnificus likes things to be pretty fresh. They like salinities between 5 and 15 parts per thousand. Vibrio parahemolyticus likes things a little bit more in the estuarine range, between 10 and 25 parts per thousand salinity. And our Vibrio vulnificus are the most virulent of the Vibrios in our region, and they do cause the necrotizing fasciitis, the flesh-eating kind of symptoms that we hear about in the news. And our Vibrio parahemolyticus can also be pathogenic, and they can cause severe gastrointestinal infections, and they're the most common Vibrio in our system. So with Vibrios, like I said, they're fairly common, and because they're so large, they can actually be influenced by the phytoplankton and the planktonic communities around them, particularly through this idea of substrate preference. So that's where they fit into our phytoplankton narrative. Vibrio tend to be associated with substrates, particularly sediments, so they like to hang on to little bits of sand and clay that can be suspended in the water. They also associate with phytoplankton and zooplankton. And all of these factors can be influenced by abiotics, so our salinity, our temperature, and then our nutrients that are input into systems. So for Vibrios, they can be associated with our phytoplankton and our zooplankton communities through two mechanisms. One is known as a phycosphere, So this is where our vibrios will essentially live in close association with phytoplankton, because planktonic cells, oftentimes they leach carbon into the water around them. It's just kind of an overflow mechanism. And that's essentially free food for our vibrios. So they will just kind of hang out around the phytoplankton because it's free food. And so there can be a close association between the vibrio and phytoplankton species. And vibrios are also known to secrete chitinases. So it's a class of enzymes that will allow them to permanently bind to chitin. So they can permanently attach and hitch a ride on our copepods and even our larval crabs and shrimp. And these can ultimately move them through the food web. So to test this, I had 10 sites in the Mississippi Sound, and I was lucky enough to go sampling with the Alabama Department of Environmental Management. We had these 10 sites, some in the northern part of Mississippi Sound, the middle part, and also south of Dauphin Island. These were sampled in 2019 from April to October, and these 10 sites represented a range in temperature from 20 to 31 degrees Celsius and a salinity range from 4.8 to 32.7 parts per thousand. So we expected the northern parts of our sampling range to have the lowest salinities overall, moving into a gradient where we had our higher salinities south of the island. Overall, we had some pretty interesting results. Using some of our extreme analysis, I looked at the highest concentrations of certain species of phytoplankton. So we identified 3 regimes. In the northern part of our sampling sites, we had a regime dominated by Akashiwa sanguinea. And the phytoplankton species that were most commonly associated with this Akashiwa were Heterocapsa and Protoparidinium. This had our lowest salinity, around 10 parts per thousand. And in this site, we found higher concentrations of vibrio, of our Vibrio vulnificus, than we had Vibrio parahemolyticus. In the central Mississippi Sound, we had a regime that was dominated by Prorocentrum micans, which is another dinoflagellate. This had a median salinity around 22 parts per thousand. It was associated with Serratium hircus and Prorocentrum scutellum. And at this location and this time, we found higher concentrations of Vibrio parahemolyticus than Vibrio vulnificus. And last but not least, we had our third regime dominated by Pseudonychia diatoms in September. This had our highest salinity at 29 parts per thousand, and these were associated with Pyrophacus horologium and Prorocentrum scutellum. And at this site, we didn't really see vibrios of either species, which we kind of figured because it was a higher salinity. And so this conceptual diagram kind of demonstrates the ideas that we're studying in this project. Ultimately, a gradient from north to south throughout the bay. We expect a proximity to freshwater inputs in the northern sites result in a lower salinity overall, higher turbidity, higher nutrients, and we expect Vibrio vulnificus to be kind of concentrated in those sites. Moving south, we expect salinity to increase with having lower turbidity and lower nutrients, and we expect Vibrio parahemolyticus concentrations to be more prevalent in this kind of mid-range. And ultimately, this gradient will affect the phytoplankton that are present in these different areas, and then that can help to structure the phytoplankton and zooplankton communities overall. And for my FDA DISL grant, I would like to give a little bit of an update. This has been kind of a crazy year so far, but I did present a poster at the virtual Southeastern Biogeochemistry Symposium in March. That was a really fun experience, getting to meet a lot of different people, even though it was virtual. And I guess the biggest news is that the oysters had to be cut from my project due to COVID and Hurricane Sally delays, just one thing after another, unfortunately, with 2020. And the plankton filters that I collected last year will be sent out for sequencing for the community analysis in the coming months. And with that, I will give control over to Allie. Maybe.
All right. I believe you can see my screen now.
Mm-hmm.
Okay. And you can see the presentation now. Ooh. probably go back to the beginning then, shouldn't we? Yeah, so thank you, Blair, for that great introduction to phytoplankton. We're going to continue along the same vein, but we're going to move into the elements and with sulfur. So maybe, there we go. Like Blair said, these processes that I'll be talking about are nutrient and elemental cycling, and so they are on longer temporal scales than the processes that Blair was talking about. These are the members of the Sulfur Squad, as I like to call us, as Jeff introduced us, and I will be jumping back between the four of our research. To get us started, I'm going to show you an image that might be a little concerning for some of you. This is the periodic table. You might not have seen it in a while, and what we care about on this particular image is sulfur. Sulfur is the 10th most abundant non-metal element found. There are 23 different isotopes, of which 4 are stable. The most abundant isotope is sulfur 32. About 95% of the sulfur is found in that isotope. There are 4 radioactive forms. The longest lived is sulfur 35, and so a lot of people do radio tracer experiments using that form of sulfur. You guys probably don't think about that. When I say sulfur, what you might think about are things like rotten eggs, maybe volcanic emissions, or those of deep sea hydrothermal vents. Or if you're a pure chemist, you might think of pure sulfur. But what we care about is sort of the global sulfur cycle and how that influences the biological system. So this is an overview of the cycle. You can tell that it's pretty complex. You know, we have these terrestrial influences, you know, there's weathering and whatnot. There's also a lot of anthropogenic fossil fuel burning emissions of sulfur into the atmosphere. And these all eventually make their way into the system that we care about, the marine system, in the form of sulfates most often. What we care about are also the organic sulfur compounds that are biologically produced in the marine environment. If you guys just need a quick refresher, these are our compounds that we tend to care about the most. So the biggest one is known as dimethylsulfonneopropionate. That's a mouthful, so we usually just call it DMSP. It is, it's an important sulfur compound produced by phytoplankton and other plant species. We also care about S-methylmethionine, or SMM. That is a precursor to DMSP. And we also care about the degradation products dimethyl sulfide, or DMS, and methanethiol, or mesh. And you can see all of their structures there on the right. And we look through, we look at the marine system through the lens of the cycling of these sulfur compounds. So this is a quick general overview of the marine DMSP cycle. And where it starts is here on the right with the production of DMSP by phytoplankton. And phytoplankton produce DMSP at a variety of levels, and we can actually split these into 2 different groups. So we have what we call the high producers. So these are phytoplankton that have intracellular DMSP concentrations between 100 and 500 millimolar. So these are your things like your coccolithophores or your dinoflagellates. We also have a grouping known as low producers, so they have intracellular concentrations below 100 millimolar. And unfortunately, these are your things like your diatoms, sorry, Jeff. And then there's also non-producers. And so we study how and why these phytoplankton are producing DMSP. And so we start with Ali Rellinger's work, and she studies Phaeocystis and Phaeocystis antarctica in particular, which is a high producer of DMSP. And so she is the one who did her research in the Ross Sea. I think she went about 10 years ago on a cruise. And what she found was that she was looking at single cell versus colonial colonies of Phaeocystis. And she found that both groups tend to produce about the same amount of DMSP. So if you look here on this bottom graph, so they have about the same contribution to the marine DMSP cycle, those 2 groups do. She also found that the light level, so that's the high versus low light in these boxes, the white versus gray boxes, She found that the light level did not have a big influence on how much DMSP was produced by these organisms. In her final research chapter, she looked into the retention of DMSP in Physostis, and she found that they are able to maintain their intracellular concentration on the order of months over the dark cycle in the Antarctic winter, which is really cool. I also look into the production of DMSP. So my plan is to look into why they are making DMSP. There are some established reasons that are stress-related responses. So DMSP is a cryoprotectant, so that's against cold temperatures, an osmoprotectant against salinity stress, and even a grazing deterrent. A recent hypothesis has come out that phytoplankton may be using DMSP as a way to balance growth during times of high energy stress. And so we've done some preliminary experiments to try and see if we can test out this theory using different phytoplankton groups. So I have looked at high producers, which are the ehux and the Gambray discus on this graph, and T. rotula, which is the low producer. And what I found so far is that I can modify the DMSP demand by these phytoplankton and actually stimulate the uptake by all producers. And so what we're hoping to do next is look into the degradation to see if, at these times of unbalanced growth, if they are using DMSP as a way to conserve things like nitrogen and phosphorus in the cells instead of sort of wasting those in the production of products they might not need. We also look into how DMSP is synthesized. So this is Kyle Hostas' work. You might have caught his defense a couple weeks ago. I won't get too bogged down into the details of the synthesis pathways. What Kyle was focusing on was Gambray discus again, and what he wanted to know is which pathway Gambray discus uses to produce DMSP. And his hypothesis is that they are using SMM on the top left there to produce DMSP. And so he developed a method to sample SMM in liquid samples using a high performance liquid chromatography or HPLC. And so he was able to develop that method and test it on a variety of different plant and algae samples. And what he found is that he was able to detect SMM in higher plants, things like sugarcane, but was unable to detect it in these algae. That doesn't mean that the SMM isn't there, it just means that this particular method couldn't detect it. Once phytoplankton have produced DMSP, they usually release it into the water column through a variety of processes, things like grazing, viral lysis, leakage, or even cellular death. At this point, we term it as dissolved DMSP, and it's available for other organisms in the water column, such as bacteria. Bacteria really love DMSP. It's an excellent source of reduced sulfur. It can actually supply up to 100% of their sulfur demand. So they take it up quite readily. And that's where the other half of our work is focused on the degradation of DMSP. One of my big chapters looks at this in particular. So bacteria can process DMSP through two different pathways. There's the green pathway known as the demethylation pathway, in which they produce methane thiol and ultimately assimilate the sulfur into their protein biomass. If they don't assimilate it, they can cleave DMSP using a lyase enzyme into DMS. That is the climatically active gas that Jeff was talking about earlier. It can help form clouds in the atmosphere and have an overall cooling effect on our climate. And so I am investigating the conditions that lead either to assimilation or DMS release from these cells, from these bacterial cells. And so I've done some preliminary experiments using water samples from the Sea Lab dock. And I've been working on the hypothesis that it's more or less the bacterial cell's sulfur demand that drives whether they demethylate or cleave DMSP. And so I've done some preliminary radio tracer experiments to investigate this. So I'll show you 3 separate experimental days. To orient you, on the top, it's going to always be bacterial production. This is a proxy for sulfur demand. The higher bacterial production, the higher sulfur demand of these cells. The bottom left is what we call the assimilation yield. This is the relative amount of sulfur from DMSP that's being assimilated into their biomass. And on the bottom right is DMS yield, or the relative amount of DMSP that's being produced into DMS. And so what we found is that we can always stimulate bacterial production using this glucose and nutrient stimulation. So we can increase our sulfur demands, but that doesn't always have the same response in our samples. So there are times, like on this particular day, where our hypothesis seems to be leading us in the right direction. So we have this increase in sulfur demand, and we have a corresponding increase in the amount that they are assimilating into their biomass, and a decrease in the amount of DMS that they're producing. On other days, we've seen slight decreases in the amount that they're assimilating with no change in the amount of DMS that they produce. And on even wilder days, there's essentially no significant change in the amount that they assimilate, but they've actually increased the amount of DMS that they're producing. And so what we're thinking now is that, yes, the sulfur requirements of the cells are probably the main driving force, but there are also these other factors that are contributing, things like physical factors and probably food web dynamics. And so I will continue these experiments to continue investigating the relationship between these. Our final work from Tara Williams focuses on the compound methane thiol itself. So this is a known degradation product of DMSP, like I've said, but it's understudied in the literature outside of sort of the assimilation aspect. And so Tara was looking into sort of the possible sources of methane thiol, so precursors in the environment, as well as what can happen to it. And so she did this on 2 different cruises in the subarctic Pacific in 2016 and 2017. And what she found, if you look at these top graphs, on the x-axis is the total amount of DMSP, and on the y-axis is the amount of methane thiol that she measured. The closest relationship is between the total DMSP and the methane thiol. There's also a pretty strong relationship between DMS and methane thiol, but that's likely a result of the degradation of DMSP itself. She did some addition experiments as well to test if there are any other precursors besides DMSP. What she has found is that DMSP is the only one that you measure that she has measured in the field, but it is likely still the primary precursor to methane thiol. There are other compounds like tryptone and casamino acids that she has confirmed are precursors. And then other things like SMM and DMSA that might be potential precursors. And she needs to do some more investigations into that to confirm. She did, as well as these precursor experiments, she did some photooxidation experiments, and she found that methane thiol is photochemically active, which means that it can be oxidized by UV radiation and consumed in the water column or in the atmosphere that way. And finally, she did some calculations on the flux to find the relative amount of methane thiol produced from the ocean relative to the amount of DMS produced. And she found that the flux out of the ocean of methane thiol is about 10% conservatively that of DMS. So it is a potentially big component of the sulfate aerosol system and the cloud formation that we've discussed. I just spit a lot of information at you, so I'm going to take a step back and we're going to look at the bigger picture here and why this is important. So a few years ago, I think it's 6 years now, the Intergovernmental Panel on Climate Change released their report, and in it they had this radiative forcing budget. And this is just sort of an overall look of the components that are affecting climate change in our atmosphere. And what you'll notice on this is that, you know, things like carbon dioxide and methane that we know are greenhouse gases, you know, we have a very high level of confidence about how they're interacting in the atmosphere and what they're doing. Where our work influences is on the negative radiative forcing, on the cooling effects that these sulfur aerosols can have. And it's important for us to continue this work because we have low confidence in how these components are affecting our climate system in general. And so we need a better understanding of that to better inform our models to get a better understanding of how climate change is gonna change in the future. That's all I have. If you have any questions, feel free to ask them at the end, but for now, I'm going to kick it back over to Jeff.
All right, so I'm going to finish it off with a little bit about our silicon cycling. 3 of us involved with that, myself, Becca, and Ryan Roseborough, who's new to the lab. This is a map of surface concentrations of silicon in the ocean, and it's got a pretty substantial range from the open ocean, which can be well below a micromolar or about a micromolar, up to 50 micromoles per liter in the surface ocean of the Southern Ocean and some places in the Bering Sea and subarctic Pacific. And then if you want to think about things in the sediments, these are pore water profiles on the plot on the right that show how silicon changes in the pore water of sediments. So instead of thinking about the surface waters, we're just thinking about the surface 25 or 30 centimeters of sediment. The interesting thing about pore waters is that typically where this concentration's asymptote is still well below solubility. That means if all the diatom silica or all the silica in the sediments dissolved, it should be at that solubility line, but there are processes which never allow it to get there. And so the questions I have basically is, how does silicon affect diatom contribution to primary production and oceanic carbon sequestration in the water? And then in the sediments, it's what are the processes that are controlling the dissolution of their silica and lead to this consistent undersaturation in sediments? And their relevance is actually to similar processes. So globally, we want to understand diatom's contribution to primary production because diatoms are a large phytoplankton group, and usually if an organism can consume a large plant or a large phytoplankton, that's time that less time they have to search for other things, and that can sustain higher trophic level biomass. So most of your dominant fisheries in the world are in systems that are typically diatom dominated. Think the Bering Sea, river plumes in the Gulf of Mexico, the Peruvian upwelling system. And of course, this has relevance to the CO2 in the atmosphere because primary production can move CO2 from the atmosphere to the deep ocean and get it out. Now, the interesting thing about the silicon sequestration in the sediments is this also has a feedback on diatom productivity. If silicon dissolves in the marine pore waters, it can come back into the water column and fuel additional diatom productivity, which can in turn also fuel more CO2 drawdown. However, some of the other processes that we've talked about work against that. If silicon can stay in the pore waters of these sediments through these reverse weathering reactions, that actually can produce CO2. So these processes, while one's in the water column and one's at the sediment, they have relevance to the same kind of global scale processes. And so one of the places we've looked at this in the past couple of years has been in the Arctic. And the Arctic has a spring bloom period that is pretty immense. There's a rapid drawdown in CO2 in the waters, and it's facilitated by diatoms. Diatoms, the PCO2 in these data, this is a database, a global database of Arctic data published by Takahashi et al. In 2009. And the correlation of silicon to PCO2 is really strong and really significant, but not so much to nitrate, which implies that there's a role of silicon in the Arctic with helping to control the amount of PCO2 that is drawn down by diatoms. So how will this change as climate warms? We don't really know. What about, well, if you increase the silicon from glacial melt, because land-based glaciers have a ton of silicon in the water, and when they erode the rock, that also releases a lot of silicon into the ocean. What are the food web implications of a changing Arctic? And then what about the ice diatoms as the Arctic changes? And we've actually combined, with the Mosaic expedition, which I talked about earlier, we're going to be looking at that and understanding how diatom biomass changes in the ice cores over a year. But thinking more about this spring bloom period, we've had two recent studies, one in 2018 and 2019, talking about this. And the first paper in 2018 was in Svalbard, which is north of Norway, top of the world. And we noticed that diatom's contribution to primary production is kind of a bloom and bust. At 4 out of our 6 stations where we were able to make these types of measurements through comparison of diatom rates and community rates, they did anywhere from 48 to 100% of all the primary production. But then we got to a couple of stations that were influenced by the relatively warmer waters of the Atlantic, where diatoms were only about 5% of the primary production. And so the reasons for this variability are unclear, but these are things that we're exploring in my lab. Moving to a study that we did in Greenland, we actually were able to go to one site and study the progression of the spring bloom. And this is somewhat of a busy figure, but you can see in the upper panel A, the blue, how PCO2 is drawn down as the bloom progresses. As you see this increase in the green chlorophyll, the PCO2 goes down. If you go to the lower figure, you'll notice that as silicon, which is I put on the line here as red, as that gets drawn down, that's really strongly correlated with the drawdown of PCO2 and the uptick in diatom biomass. When we actually did rate incubations in each of these cruises to understand what the role of silicon was, in Norway, in the Svalbard, we found that a lot of the samples, the silicon was so low that they could actually be growth limited by it. And the reason that they're growth limited is by the amount of plasticity that they have to change their cellular composition. It's just not enough to overcome these exceptionally low concentrations. And when we added silicon to samples in Greenland, we noticed that the growth rate was stimulated towards the end of the bloom as silicon was running out, showing that they are yield limited by the quantity of silicon that is available. Because what's amazing about this system is there's plenty of nitrate left over. So when diatoms basically tank and get limited by silicon, there's still nitrate left over for other phytoplankton groups to use, and then that kind of fuels a successive bloom that will extend into the late summer. But it's not a really pulsed quantity of primary production that a diatom bloom gives. So just to not saturate you too much, we're going to just close things out with the sediment work. And this is some data from 2016 and 2017 during what we call the classic cruises. This is part of Rebecca Pickering's dissertation. We did this work on the RV Pelican using an 8-spot multi-core, and then you can see how some of the sediment cores that we were reusing. And Rebecca reported data from all these stations, but specifically what I'm going to show you relates to these 3 stations. And one of the things that's interesting about river plume sediments is that when you digest sediment for diatom silica, you get a value. But in river plume sediments, diatom silica is usually coated by minerals and precipitates, stuff that's being leached out and dumped out into the rivers and being precipitated through chemical processes. And so these traditional methods aren't really good at catching diatom silica that's been altered that way. So we take it through a stepwise approach of sequential extractions using acids to break down some of these precipitates, using bases to digest the silica, and the silica that comes out is usually significantly higher than using a traditional method. And then we use a stronger base to get a little bit at this refractory silica, things like sponge spicules, or even some of the clay minerals. And the nice thing about doing this in this environment is we were able to look at stable isotope pools, which give us a fingerprint of what types of silicon these pools are derived from. So this is DEL notation, and I'm going to simplify this, showing that everything to the right is isotopically heavy, and everything to the left is isotopically light. And those pools that traditionally get at diatom biomass show isotopically heavy silicon. And what that tells us is that silicon came from the water column, where diatoms drew the silicon concentration down a lot, and the remaining silicon in the water was isotopically heavier because diatoms would preferentially use the lighter isotope. But when we look at these other pools, specifically the authogenic pool that leached material, those precipitates, these are some of the most isotopically light silicon values ever reported in the ocean, which implies that in order for that to happen, you have to have a lot of cycling and kind of selectivity of these lighter isotopes by chemical reactions. And then this lithogenic pool is kind of sitting in the middle. This is a very large pool, but it looks very similar to something you might expect in clay, because the standard for these isotopes is a terrestrial reference quartz. And so what this allowed us to do is kind of look at the differences between these pools and ask some questions. I won't get into those much, but what I want to get into is just the difference between the old method and the revised method. I mentioned that when we add these leach protocols, we extract more silicon from the sediments because we're accessing a pool of diatoms that has not been coated by minerals. And that excess in this figure is called just an apparent alkaline. This is just an excess that we get back by changing the method. When we look at the stable isotope composition of that, what that tells us is it's really heavy. And this is important because when we look at pictures of diatoms in sediments, we see a lot of pictures like this where they're coated by these minerals, or they have all these flecks and flakes on them. And these SEM images are telling us that diatoms are being coated, but the problem is you're looking at one particle in trillions, and you can't really infer that much from a couple particles. What this bulk analysis tells us with the stable isotopes is the pool that's released, that extra silica that's released, it's isotopically heavy. And isotopically heavy silica in a pool in the sediments is only going to be coming from water column diatoms raining down to it. So this is the most kind of robust bulk measurement that we have to say, yeah, diatoms in these sediments are getting coated in these clays, these reverse weathering reactions, which ended up producing CO2. And this is a new way forward. So just kind of finishing it off, the next steps, this is where Ryan's coming in, is these are the same data from the same cruises, just we're adding some metals. And you can see that in these different kind of traditional versus sequential leaches, this orange is these kind of metal leaches that get rid of a lot of these precipitates. The precipitates are heavy in iron and manganese, and the titanium data tells us that the precipitates are likely of marine origin. They're not coming from a terrestrial environment. And you can see these strong differences that are very similar to what I showed in the last figures between the kind of the clay-like fraction, these authogenic silicas, and the diatoms. And where we're going next with this, with the help of Dr. Shang Lang, is Ryan will be looking into lithium. And lithium is another isotope system that you can use to understand these kind of silicon reverse weathering processes. You can see just from these pilot data that there are some differences between these pools, and the reference lines, lithium and seawater. So these lithium extracts don't look anything like lithium and seawater, and they look like a combination between continental crust and secondary clays, which are those precipitates that are forming. So what we're hoping to do with Ryan's work is use lithium and silicon isotopes to kind of constrain the process in general. Thank you for your patience, but before you ask questions, we want to know, have you thanked the phytoplankton today? And thanks also for your indulgence. It's been a pleasure, and if anyone has any questions, please feel free to jump out and ask them. You can also list them in the chat window and I can read them out. Thank you. All right, we stunned everybody.
Hi, Jeff. There is a question by Bill in the chat window.
What does a shirt cost? Blair, what was our cost on those?
I think 15.
We'll give you at cost. Blair did a nice custom color design on 2 sides. So send me your size preference, and yeah. So Bill's question for Blair: were you able to process any of the oysters? And he's interested to hear if you got any preliminary results before Hurricane Sally.
So yeah, that's a good question, Bill. So I sampled oysters. I think I got 3 samplings in before Sally, but that was doing for like basically my plan B, which would be DNA extractions for metagenomic analysis. I do have those oyster extracts, but I haven't really done anything with them in terms of metagenomics yet. And then far before that, I have oyster extracts from November through March, like last November through March, but those are sitting at FDA. Yeah, so.
And FDA is closed, right?
Yeah. Mm-hmm.
Okay, so another question from John Leder. Has anyone put the CO2 taken up or produced by weathering and reverse weathering into the CO2 budget for bottomizers? Seems like it might be an important sediment water exchange of CO2. Right now, we just kind of got a handle on the global magnitude of reverse weathering in the last 10 years. The most recent, and I think first true global estimate of reverse weathering was in 2017 by Shirley Rahman, who actually just gave a seminar at USM because she's a postdoc there. But the magnitude of that is on the order of kind of 3 to 7 teramoles of silicon, and that's basically what the rivers dump in to the ocean. There's other sources, hydrothermal vents, but in terms of the CO2, that's a kind of a big question, and I think it's an important one, especially in a lot of these lower latitude river plume systems. And so I've had a discussion with somebody at Texas A&M on this, but we've never really followed it up with any action. It would be something to talk more about. All right. Well, thank you, everybody. And if you guys have any questions about the projects, I encourage you to go talk to the students. And if you have any other questions for me, please email or come see me when I have an office again. Thank you very much, and we'll talk to you guys later.